was asked to talk about biomarkers in clinical studies, and I have an example of a biomarker that did go the whole way from research collaboration and to FDA approval, and I will try to explain to you this story. I was also asked to tell something about the number of patients and samples that can prove statistical power, and I have a simple answer to that. You cannot have a rule. It depends upon the specific clinical question, the medical need, the studies you have, how good is the response, how many patients respond. There's a lot of issues, so it's not possible to answer this. It also depends upon how to use the marker. Is it a screening marker? Is it a diagnostic, prognostic, or predictive marker that you are going to study? So I can give you one good advice. Find a very good biostatistician and make this one connected to the project and make the calculations. Don't do it yourself. So every day there's several cancer biomarker studies that are being published, and very, very few biomarkers are available for the clinic. Why is that? It is because most of the studies is studies of convenience. You have some sample in the freezer and you have an experimental method, and then you combine it and you publish it. And I know that EU have several good biomarker experts that are discussing for the moment to get to the editors of the different journals and convince them not to publish so much of these studies that are more noise than even something that you can use. So because of this convenience, these types of studies are very unstable and unlikely to, or likely to be of chance. Therefore, one of the very good biostatisticians has published a paper about the level of evidence, and Niels mentioned it in his presentation. That level, the highest level, is where you have a prospective randomized clinical trial where you randomize the patients according to the biomarker. These studies are very, very rare. I cannot even find one example. So what you normally will do is that you make a planning prospectively on a retrospective study that has been conducted where you are able to find the samples. And I will give you an example of this. Level 2B is if you don't have any validation of this one, or you have inconsistent results. And then you go down, it's patients not in a clinical randomized study, but standard of care, and you have no prospective planning, you are at level 4 and 5. So why are you getting these inconsistent studies and results? Very often it's because you don't use validated methods, you have variation in scoring criteria, in the cutoffs, patient populations, and the treatments that you are comparing. And I have identified two of the most common mistakes made in this area. One is that people think they can make a predictive marker, but they don't have a really control arm. And the other one is that people are using the median cut point for a biomarker, and then you divide your population into two halves that are of equal size, so the statistics becomes very good. But when you try to validate this in another study, it would be another cut point, and you will never reach a point where you have a biomarker that can be used in the clinic. So it's only for exploratory studies that you can use the median cut point. This is the same figure as Niels has. Here you have a patient population, and you want to either find a predictive or a prognostic marker. There's some of the patients have good survival, some have poor survival. Some have poor survival, but the survival improves if you get the right treatment. So the question is, test the patient population and find out in what group your patient is. And I have some yellows because there's always somebody that doesn't fit into anything. Here we have some examples of prognostic and predictive markers. Here is a prognostic marker, and there's no benefit of therapy. You have good survival if the marker is negative, poor survival if it's positive, but there's no change whether you are in the control arm or in the treatment arm. Here you have a prognostic marker. You have a benefit of treatment, but it's the same in both groups. And this one can be tricky. If you don't have the right control arm, you will not be able to find this difference, and perhaps you think you have a predictive marker. Up here you have a predictive marker, and there's a benefit if the marker is positive. Here we have the same in the control arm. You have a poor survival, and it increases if you get the right treatment. And what is the most common thing is that you have a combination of both prognostic and predictive effect. So you need to do studies that you can find out whether what is the component that is predictive and what component is prognostic. Oh. Here's 2 examples from real life in breast cancer. One is the estrogen receptor. You have if it's negative, you have a poor survival, a little bit better if it's positive. And if you get tamoxifen or any anti-estrogen treatment, you will increase the survival of patients with a positive marker. And over here, it's from the top 2A, where you, in the control arm, have a very poor survival if you are positive, and it increases to the same level as the negative if you get anthracyclines. So now we come to the definitions of targeted and personalized medicine. Targeted treatment is if you have a target for your drug. And a very good example is Avastin. It targets VEGF-A, but no biomarker is available, so you cannot call it personalized medicine. One example where you really have personalized medicine is if you have Herceptin. It targets the HER2, and the biomarker that stratifies the patients is the same marker, the HER2. And then the old way of treating, one size fits all, where you neither know the real target or have a biomarker. And I think that most of the chemotherapy that are used today is of this in this class. So there's a demand for development of FDA-approved and CE-labeled biomarkers that have a clinical validation. And we have tried to make such a biomarker by making the top 2A FISH PharmDX kit in Dargo. We had a drug. It was anthracyclines. The target was topoisomerase 2 alpha. The biomarker was the gene that codes for the target. And we did a validated assay. We made analytical validation, it's the first step. We made clinical validation, and for the moment, and it's not finalized yet, we have a study going on, a prospective study going on to see the benefit and to find the clinical utility. We initiated the development in 2001 by a collaboration between the Danish Breast Cancer Cooperative Group and Dargo. We made a prototype kit and a pilot study on 120 patients. Then we got to a main study that had 773 patients randomized. It was published in 2005. It was CE labeled in 2007, FDA approved in 2008. And based on all the good comments we got from FDA, we made published an update in 2008. So if we take the first part of it, the research collaboration, where we discussed how to collaborate, and we at DARCO started to make the kit. And we made research use only kit, but it was in a format that was locked very early on. And it's important to lock the test, because otherwise you have to repeat the results you later get. The benefit from both parts was 3 patent applications, numerous oral presentations and 4 posters, 12 publications, and 1 PhD. The product development, we started the research use only kit development in 2001. We started using it in the research collaboration in 2002, and it was launched in 2003. And basically, it's the same kit that still exists today. It was used first for the pilot study, and then it was used for validation on normal breast tissue and on cell lines. The kit measures gene copy number of top2A, like the one Niels mentioned. We have a centromere probe as a reference probe, and the yellow one is for the gene. We developed it so it could score both amplifications and deletions, and it has a predictive benefit for anthracyclines and is also a very strong prognostic biomarker. The gene codes for the enzyme topoisomerase 2 alpha, and it's the primary target for the anthracyclines. Around 80% of breast cancer patients have normal level, 10% have deletions, and 10% amplifications. And again, those that have aberrations are likely to have benefit from anthracyclines. Then it comes to a clinical validation, and in parallel we made a kit for the HER2, and it had higher priority, so we finalized that in 2 years. But there was an existing kit on the market, so we just had to make a comparison to the existing one. Therefore, we only needed 250 patient samples. Later on, we had to have some Japanese patient samples because they have a specific approval process in Japan. So in fact, we got the study published before we got the CE labeling and the FDA approval. It took 7 years to get it approved by FDA, and there was 773 patient sample. I don't know if you should say required, but that was available. And we expect next year to have the clinical utility ready. Here's the study. There was 980 patients included. We have a control arm without anthracycline and the test arm with anthracycline. And it was a very good study, that it was well designed, and we made a prospective plan where we locked everything, the kit, the scoring method, the cutoff, and so on, up front. And we had a very long observation follow-up time, 10 years. Here's just the results. We had approximately 10% of the deletions and amplifications among the 700 patients 773 patients we were able to get tissue from. There was, of course, some patient we couldn't get the tissue on, and we had one specific pathology department. All the samples was unsuccessful because they had a specific way of fixation, and we couldn't make fish on those. Here we see all patients in the study, so we have the prognostic value and patients with a normal status have the best survival, and those with deletions have the worst survival, and amplifications is in between. Another way to look at the data is to divide them into the two different treatment arms. And over here we see the control arm, where patients with a normal status have this survival, and the deletions and the amplification have poor survival because they don't get anthracyclines. But here, over here in the anthracycline arm, they are lifted up almost to the normal level, or to the level of the patients with normal status, because they have benefit from the treatment.
Can I just say, I think for everybody, look at the p-value at the right slide. What is it, 0.13?
Yeah.
So it's insignificant. So if you didn't have the control group at the left side, you would totally misinterpret these data. Totally.
Yeah. Yeah. Another way of looking at it is to take the different groups according to the biomarker. And here we have the two treatment arms and all the patients that have normal status. And you can see you get the same survival in the control arm and in the anthracycline arm. Here we have the amplified patients, and those patients getting anthracycline have better survival than those in the control arm. And the same case here for the deletions, but both curves are much lower than up here because they have a very poor prognosis. Another way of looking at it is in a forest plot, and you can see here that patients who have abnormal status, here we have both deletions and amplifications, have benefit from anthracyclines, and patients with normal status are almost at the level 1. We also did it for HER2, and as you can see here, the HER2 is not capable or able to discriminate between the two groups in a significant way. When you have finalized your own study and you get it FDA approved, you have to wait and see whether there will be other studies around the world that can support your own study. And we got support for supportive evidence from a Canadian study, so our level of evidence increased to 1B. Then there came some inconsistent result from a UK trial, so it was back to level 2B. I can tell you one thing. They didn't have a biostatistician. They had a technician taking a course in statistics, and I'm not sure that it could be the reason. Then there was an indirect supportive evidence from a US study from Slayman's group, but they had a different study design, so you could not really use it as supportive evidence. But at last there was a meta-analysis where both the Canadian, the UK and the Danish study and the Belgian study was merged into a meta-analysis. And it actually supported the findings we were the first to get, and later on from the Canadian group. But there was lack of scientific acceptance, even if the meta-analysis support previous results. The researchers could not really agree upon how to treat the patient, and especially people have a tendency to believe in their own results. So the UK group was not very supportive for changing their way of looking at it. I just shortly will show you some of the results. Here is the Canadian study. There was 710 patients totally, completely the same design of the clinical trial. They were only able to get 438 patients, but as they could look at our way of analyzing this, the study, and they merged the deletions and the amplifications from the very start, they were able to get a better result, even if they had fewer patients. they got almost the same, although they had a little bit different criteria for deletions, so they got fewer deletions. And here's the result. You can see that for those that are amplified or deleted, you have this difference between a patient having anthracyclines and patients in the control arm. And over here you have all the patients with normal status, and they have the same survival. Here's the meta-analysis. Don't try to get all the details. What I want to show you is that the merged data from all 4 trials is at the same level as the one we found in the Danish study and in the Canadian study. And what is really on the wrong side here with a very, very big confidence interval is the Belgian study and the UK study. Go here. This is another way of looking at the timeline. We started in 2001, and we expect the clinical utility in 2014. Up in the upper part, we have the scientific part, all the publications along the line. And down here, we have the assay development and the regulatory status. So we had kit launched in 2003, and we got PMA on the HER2 part in 2005, then in 7 and 8 on the top 2A part. And I don't think there will be any more approvals.
Can I ask you a question here?
Yeah.
So the kit was approved both in Europe and the US
Yeah.
some while ago. Do you know how much has been accepted or adopted? I mean, you said you're waiting for the clinical acceptance, but what does that mean in terms of penetration, if I may use that word, of the kit in clinical practice?
I think that because there was 4 groups having each their own kit and believing only in their own results, there's been a lot of disagreement upon how to do it. And the Americans want to do it with a competitor kit. So there's not been, even if the DARGO kit is the only one with an FDA approval, there's not been acceptance that this is the one to use.
But the FDA approval is not for prediction, it's for prognosis.
For prognosis, that's correct. Yeah, but at least there's guidance for scoring and for cutoffs and for the technical part of it, and people don't even agree upon that. They do it their own way.
So what, I'm a marketing person, so maybe that's why I'm asking this question. No, it's very interesting, and I'm just thinking about launching. I never launched a diagnostic, but one of my questions would be, how much have you included the clinicians in that development? Because they are the ones that have to adopt the kits at the end of the day.
I think that was what our marketing did wrong, was that they didn't really go to all these key opinion leaders to get them involved from the very beginning. I was the product leader all those years in the development, but each year I had a new marketing person. And they started from scratch, and I had to just tell them, how is this kit working?
Yeah. So I think it comes back to your comment earlier that it's important to have your clinicians on board very early on when you develop a biomarker.
Yeah.
Sorry for interrupting.
This is just the conclusions. We were able to prove the prognostic value and also the predictive value if you combine the deletions and amplifications. And we also were able to prove that we could not use HER2 instead of top 2A for neither the prognostic nor the predictive value. And some lessons to learn from this: use validated methods. Lock the kit very early on, because otherwise you will have to repeat your studies, and samples are very, very costly to get because there's not very many studies around that are suited. Use identical cutoff values if you are comparing, and standardized scoring of signals and everything is very important. We had some issues with the tissue pretreatment, we could actually turn a amplified sample into a normal sample and a deleted into a normal sample by changing some of the pretreatment conditions. That's the same case with immunohistochemistry, where you can, by changing the pretreatment, change the status of the tissue and of the patient. So beware of that. Also find the right patient sample from the right clinical studies, randomized studies with a control arm. Be aware that you cannot compare different treatment regimens. And in this case, we could not compare to studies where they had a pre-selection for patients with HER2 positivity, because we also had to have those normal patients in order to prove the clinical value. So overall, we can use retrospective studies with a proper design to make biomarkers. So, and I recommend that you go on the clinicaltrial.gov to see if there are studies around the world. There could be some studies that you were not aware of. Use validated tests. Cutoff values and scoring criteria are very crucial. The analytical and the clinical validation has been performed in this study, but we remain to have the clinical utility. We have a DBCG READ study that is ongoing, and here the patients are randomized and treated according to their top 2A status. And the first results should be available in 2014. A long way.
Thank you, Kirsten.